What are cell walls? They are generally a rigid structural layer, sometimes multiple layers outside of the cell membrane. They provide structural support for the cell. They protect the cell from the outside world. They function with the cell membrane to filter what goes in and out of the cell. They serve as a pressure vessel to keep the cell from exploding due to osmotic pressure. The background of this slide is an electron micrograph of the cellulose fibers of the green alga Penium margaritaceum. Structural carbohydrates are used to build things like cell walls. Structural carbohydrates are not usually used for energy storage. In addition to pigments and energy reserves, algae are classified by the types of structural carbohydrates and the molecules associated with the carbohydrates. But we need to remember, some algae do not use carbohydrates to build cell walls. These algae are classified by the other molecules they use to make up the cell wall. The main structural elements of algal cells are polysaccharides or sugars. Differences in the chemical composition of the polysaccharides results in different physical properties of various types of cell walls. No algal cell wall consists of only one type of molecule. It's the interactions between these molecules that produce the distinct properties of the cell walls of different algae. The main polysaccharide in most algal cell walls is cellulose. Cyanobacteria have no cellulose, and they use a sugar protein molecule called peptidoglycan. Chlorophyta, the green algae, use cellulose and proteins in their cell walls. Rhodophyta, the red algae, use cellulose plus agar or carrageenan. Some red algae use the polysaccharide xylan instead of cellulose. The Phaeophyceae, the brown algae, use cellulose and calcium alginate and mucilage for their cell walls. Cyanobacteria have cell walls made of peptidoglycan. Peptidoglycan literally means protein sugar. It is a polymer of sugars and amino acids that form the cell wall in bacteria. The sugar components are N-acetylglucosamine and N-acetylmuronic acid. These sugar monomers alternate in the chain and are linked by beta-1,4 linkages. These polysaccharides are cross-linked by specific proteins that are different for different species. This net of sugars and peptides forms a very stable cell wall that is very chemically resistant. Almost all bacteria use peptidoglycan for their cell wall. The green, red, and brown algae all have cellulose as the main component of their cell walls. Green algae also have many proteins associated with the cell wall, as well as mannan, xylan, and pectin carbohydrates. You are probably familiar with pectins as the thickening agent used in canning fruit. The red algae also have mannan polysaccharides in the cell wall, and lots of agar and carrageenan. Agar is commonly used in microbiology for pouring Petri plates and as a food thickener, especially in canned meats. Carrageenan is used in ice cream and is a vegan alternative to gelatin. In addition to cellulose, the brown algae have alginate and mucilage in the cell wall. Alginate is a very useful compound. It is used as a food thickener, for fireproofing fabrics, in lithium-ion batteries, in pharmaceuticals, and to treat burns. Mucilage is most common as glue, but it is also used as medicine and as a food additive. The haptophytes, glaucophytes, and dinoflagellates all use cellulose for the cell wall. The haptophytes also produce cellulose plates that are substrates for calcium carbonate, which you call chalk. These structures are called coccoliths, and the haptophytes that produce them are called coccolithophores. These algae form blooms in the ocean that can be seen from space and are responsible for the chalk in the cliffs of Dover in Great Britain. The glaucophytes have cellulose cell walls that are enclosed in larger mother cell walls also made of cellulose. It has been argued that this makes the glaucophytes colonial algae, and some people have used the term quasi-colonial. The dinoflagellates have cellulose plates called theca. These plates form an armor shell around the cell. Again, it should be remembered that living things can make structures that vary significantly in function and morphology from the same starting materials. Diatoms have a cell wall made of silica, or glass. They are like little canisters with a box and lid. The box and lid structure is called a frustule. In the Victorian era, microscopists made very intricate designs by placing diatoms on slides. Today, very few people still practice this art. A link to a short movie about a living practitioner is at the bottom of the slide. Cryptomonads and euglenids both have cell walls made of proteins. The cryptomonads have cell walls that are made of protein plates sandwiching the cell membrane between the outer and inner protein cell walls. Euglenids have a cell wall that's made of protein strips underneath the cell membrane. As you can see in the electron micrographs, the structures are regular, but are distinct between the two phyla. The proteins used to make these cell walls are not related to each other.
A short video of Euglena spirogyra at 1,000 times magnification by differential interference contrast microscopy clearly shows the protein strips in the pellicle of the alga.
The chrysophytes and chlorarachniophytes do not have a cell wall. The chrysophytes make chambers out of organic material that is mixed with silica called lorica. They essentially live in little glass cups. Chlorarachniophytes are really photosynthetic amoeba and move about just like any other amoeba. Their cell membrane is connected to the cytoskeleton on the interior of the cell, just like animal cells. This summary slide includes the 4 algal phyla that we've been following for the past few lectures. As you can see, as more and more traits are considered, the differences between the phyla are more and more pronounced. Is it a prokaryote or eukaryote? Does it have flagella, and if it does, what kind? Are there unique carotenoids? Are phycobilins used for light harvesting? What chlorophylls besides chlorophyll A does it use? What reserved carbohydrates are used? How are the monosaccharides linked together? Where are the carbohydrates stored? And what kind of cell wall does it use? The same comparison slide can be made for all the other algal phyla we have been looking at, but would take up several more slides. We leave that comparison to the interested student to complete on their own. Next lecture, we will look at the differences in cell structure and morphology.